Discipuli. This is Mix Martin with a recap of lessons 4 through 6. Lesson 4 was about multicellularity, and the question is, what are the advantages of multicellularity? So why would organisms be made of more than one cell? The first thing to examine is the limitations to cell size. And the question is, why are cells so small? So why are you made of trillions of tiny cells instead of a few big ones? To understand this, we need to know what diffusion is. Diffusion is the passive movement of particles from an area of high concentration to an area of low concentration. So if you think about if you're in a room and someone sprays body spray or air freshener, eventually everybody in the room is going to be able to smell it because the particles of that air freshener spread out through the air in the room. So this is something that molecules do naturally because of the random kinetic movement of molecules. Eventually, they spread out through the available space, and that spreading out is called diffusion. Diffusion, again, goes from an area of high concentration to an area of low concentration. We also need to understand the relationship between surface area and volume when something gets larger. So these are some cubes here with some simple geometry. We can figure out the surface area and the volume of each. So in our smallest cube, the surface area is 6 faces. Each face is 1 by 1, so that's 6 centimeters squared. The volume is length by height by width, so 1 times 1 times 1 is 1. So I have a surface area to volume ratio of 6 to 1. But if we look at our bigger cube, our surface area, each face is 9 square centimeters. So we have a surface area of 54, 9 times 6. And the volume is going to be 3 times 3 times 3, which is 27. So now we have a surface area to volume ratio of 2 to 1. So as size increases, surface area to volume ratio decreases. That's really important. Because larger cells have a smaller surface area to volume ratio, we end up with a problem because nutrients need to diffuse into a cell, waste products need to diffuse out of a cell, and this can only happen through the membrane of the cell. So if you have a huge cell, it's going to take so long for materials to get to the center of the cell by diffusion that cell would just not be able to be a functional living cell. So we have many small cells to minimize the diffusion distance from the membrane throughout the cell. The next section is emergent properties. So we know that multicellular organisms are made of many cells working together. You don't have to memorize this hierarchy, but it can kind of help you conceptualize multicellularity. So cells are organized into tissues with other similar cells. Tissues of various types are organized into organs, such as this heart. Organs come together to make organ systems, like the circulatory system of this horse. And then the organism is the whole horse. So this horse has many different types of cells coming together to make many different organs. And all together, all of those cells work to perform the functions of life for that horse. Emergent properties arise from the interaction of component parts. So in class, we talked about what could you do in a class that a single student can't do. And a class of students, of many students, can have a discussion, can have a seminar, can play a competitive game, can play scooter soccer. Those can be considered emergent properties because you can't do it with one student. You need interacting students. So multicellularity allows for emergent properties. Multicellular organisms are capable of completing functions that individual cells could not undertake. So because of specialization, multicellular organisms can do all of these different interesting things that a cell on its own can't do. Next section is differentiation. The question is, do all your body cells have the same DNA? The answer is yes. with the exception of your sperm or your eggs. Those are made in a process called meiosis, and they're genetically unique. But any of your body cells will have the same DNA. And I put that person swabbing the cheek to remind us that forensically, if you find blood or you find sexual fluids or you find remains, you can compare that to the DNA of a living person just by painlessly swabbing their cheek. And you'll get a few cheek cells that you can then extract DNA from. And that would only work if all the DNA in an organism was the same. So all of your cells have the same DNA. So the question is, if all of the cells have the same DNA, how come our cells are so different in structure and function? And the answer is this process called differentiation. So in differentiation, a cell becomes specialized for a specific function. Differentiation is the result of differences in gene expression. And all of your cells have the complete DNA genome. They have the whole library, or they have the whole cookbook. But at any given time, a particular cell is certainly not expressing every single protein in the genome. They're not making every protein that they have instructions for. They're going to make specific proteins depending on their role. So in this image, we have a single cell. Maybe this is an embryonic cell where the embryo is still developing. Divides into daughter cells. And then through the expression of certain genes and not others, these cells become different. So this says expression of an epithelial specific gene turns this into an epithelial cell. Expression of a neuron specific gene turns this into a neuron. So because these cells are doing transcription and translation of different genes, they're making different proteins. Proteins do almost everything in the cell. So if you have different proteins, you're going to be a different shape, you're going to be a different size, you're going to be able to perform different functions. I think this is what I just said. So if different genes are transcribed and translated, we'll have different proteins produced, which will lead to different cell properties and functions. Going on to lesson 5. Lesson 5 is about prokaryotes. And the question is, what are prokaryotes? All right, so let's start. What are prokaryotes? Prokaryotes lack a nucleus and other membrane-bound organelles. So they do not have a nucleus, and they do not have any other organelles that are surrounded by membranes. They're some of the earliest organisms to evolve on Earth. You can find them almost everywhere. In your body, you have more prokaryotic cells than you have human cells. So you have tons of bacteria colonizing your skin, colonizing your gut. Prokaryotes are everywhere that we look. Some etymology might help us remember what this means. Pro in this case is before, it's like pre. And karyon is a Greek word that means nut or kernel. The kernel that we're talking about is the nucleus. So prokaryote are organisms that evolved before the kernel, before the nucleus. And that's why I made the point about them being evolutionarily older. Prokaryotic structures. The plasma membrane regulates entry into the cell. So it has certain membrane proteins that allow entry of particular molecules, but not others. The cell wall surrounds the cell membrane. It protects the cell and maintains its shape. So prokaryotes obviously don't have skeletons, but they have the cell wall that keeps their particular rigid shape. The nucleoid is the region with the genetic material. So it's here on this image. It's not a nucleus because it doesn't have a membrane, but it's a nucleoid, and that's referring to the region where we see the genetic material. Many prokaryotes also have plasmids, which is this additional genetic material in a ring. So here's a plasmid. Prokaryotes have 70S ribosomes. And the 70S is about their size. Specifically, it's about how they separate in a centrifuge, but you don't need to know that. Just the 70S ribosomes are found in prokaryotes. They have the same function as 80S ribosomes in eukaryotes, which is translation of mRNA and protein synthesis. The pink structures here, pink and purple, are called flagella. The singular is flagellum, which means whip. When these structures beat like a whip, they propel the prokaryote forward. So if you've seen pictures of or videos of sperm swimming or animations of that, sperm also have flagella. They're different in structure because sperm are eukaryotic. But if you can imagine what that movement looks like, then you know what the flagella do. The pili are the green hair-like structures projecting from the plasma membrane, and therefore attachment. So attachment to whatever the bacteria is growing on. If the bacteria is infecting something, it might anchor itself to the tissue of the organism that it's infecting, but those are the pili. Some prokaryotes have a capsule, which is an additional layer that surrounds the cell wall, and that protects the cells in adverse conditions. So maybe if it's really cold, or the cell is in danger of drying out, or things like that, the capsule can just keep the prokaryote in a kind of dormant state until conditions improve. Prokaryotic reproduction. Prokaryotes reproduce by binary fission, which is a type of asexual reproduction. DNA has to be replicated before the cells can divide. So here we have the parent cell. The DNA is copied, so we have 2 copies of the full genome. And then those 2 daughter cells start to separate, eventually becoming 2 distinct cells. They're genetic clones of the parent cell, because the DNA was just replicated and then separated. This is showing replication of bacteria. So you can see they get longer and then divide. What you can't see is the DNA replication, but that's happening. This is also a good reminder about washing your hands. So this is how quickly the bacteria can spread out and cover whatever nutrient medium they're growing on. You need to be able to produce a labeled drawing of a prokaryote. So there's a video in the slides that you can consult. You can also consult this diagram. So we have all of the key structures represented. A couple things to keep in mind. Your flagellum and your pili, or your pilus, each pilus, I should say, should be attached to the cell membrane, which is that single line that's within the cell wall and the capsule. The nucleoid is kind of a tangle, but it is connected, so it's circular in the end. Yeah, that's what I wanted to say about this. I think the rest is fairly self-explanatory. And again, consult the video in the slides if you like. This is a mark scheme from IB, so they've asked this on a test in the past, and this is how they scored it in the past. So you can score your own to see if you drew an adequate drawing. Lesson 6 is about eukaryotes. So 5 was prokaryotes, 6 is eukaryotes. And the question is, what are eukaryotes? Eukaryotic cells have a nucleus and other membrane-bound organelles. Eu means true, and karyon again is the kernel or the nut. So this is the true kernel, meaning it's the true nucleus. So prokaryote is before the nucleus. This means it has a true nucleus. Animals, plants, fungi, and protists are all eukaryotes. Eukaryotic cells are larger and more complex than prokaryotic cells, and they rely on these small structures known as organelles to perform different tasks in the cell. So just like you have organs within your body, cells have organelles. So they're like little organs within the cell. Let's compare eukaryotes and prokaryotes. The bacterial cell here is representing prokaryotes, and then animal cells and plant cells are eukaryotic. So notice that the prokaryotic cell does not have a membrane-bound nucleus. It just has its DNA in this region called the nucleoid. Notice all of the membrane-bound organelles in the animal and plant cell, such as the mitochondria, such as the Golgi complex, the endoplasmic reticulum, and the plant cell has chloroplasts. One thing they have in common is that they have a plasma membrane. So the bacterial cell has a plasma membrane here, the animal cell has a plasma membrane, and the plant cell has a plasma membrane within the cell wall. Plasma membrane and cell membrane are synonyms, so you can say either. So eukaryotes and prokaryotes have plasma membranes. They also have the cytoplasm, which is the region of the cell outside of the nucleus for the eukaryotic cell, and it's light blue in the bacterial cell, so the cytoplasm. And they have ribosomes. Prokaryotic cells have 70S ribosomes. Eukaryotic cells have 80S ribosomes, but both types of cells need to have ribosomes because, of course, both types of cells need to do protein synthesis. This is kind of a summary table. So I said this, in eukaryotes, the nucleus is present and surrounded by the nuclear envelope. That's the membrane that surrounds the nucleus. Prokaryotes have no nucleus. They just have their DNA in the cytoplasm in a region called the nucleoid. Both eukaryotes and prokaryotes have ribosomes. Eukaryotic ribosomes are larger. The DNA in eukaryotes is in long strands that are associated with histone proteins. And histone proteins are like spools. So if you think about string that's wrapped around a spool, the histone proteins are what DNA wraps around. And that allows it to be more organized. It's not just a tangle that you don't know what's where. It's highly organized by the binding of the histone proteins. So DNA, again, is wrapped around those histone proteins, which keeps it organized and not just like a tangle of yarn. In prokaryotes, they have circular DNA that's not associated with histone proteins, and cells may also contain smaller ring-shaped plasmids. Cell walls are present in plants and fungi and absent in animals in the eukaryote category. Prokaryotes almost always have a cell wall, but there are a few exceptions. All right, last section is organelles, and this information is actually in a reading that's posted to Classroom, so I'm not going to go through it in this video because that information is available to you elsewhere. Please reach out if I can help with anything.